Hello, in this video we're going to look at the Nobel Prize in Physiology or Medicine from the year 1907, awarded to Charles Louis Alphonse Laveran, in recognition of his work on the role of played by Protozoa in causing diseases. This is a summary of his Nobel Prize lecture. Here you see some of the symptoms of the disease he was investigating, malaria, and here you see the life cycle of the parasite involved. Okay, Protozoa as causes of the diseases. That's the name of the Nobel Prize lecture given by Monsieur Laveran, and we will go to summarize. I have summarized it, so it's not complete. If you want to read the complete lecture, you can also follow the link in the description by the video. You can also see it in the end. Okay, here it goes. I explained how I was led to concern myself with the pathogenic protozoa. In 1878, I was sent to Algeria and put in charge of the department of the hospital at Bonn. A large number of my patients had malarial fever, and I was naturally led to study these fevers, of which I had only seen rare and benign forms in France. Malaria, always known in the north of Europe, but is of great importance in the south. Alongside the intermittent forms, both the continuous forms and those called malignant appear. In the tropical and subtropical regions, endemic malaria takes first place almost everywhere. Algeria has become much less unhealthy, but endemic epidemic malaria rages every year. I had the opportunity of making necropies on the patient's death from malignant fever and of studying the melanoma, black pigment in the blood of patients affected by malaria. People were still in doubt about the constancy of alteration in malaria and about the causes of the production of this pigment. I was struck especially in 1880. I discovered the edges of pigmented spherical bodies in the blood of a patient suffering from malaria, filiform elements resembling flagella, displacing the neighboring red cells. From then on, I had no more doubts of the parasitic nature of the elements which I had found. I described the principal appearance of the malarial hematozoan, which received with much skepticism. In 1879, Klebs and Thomas C. Crudelli had described Bacilla malaria, the bacillus formed in soil and water in malarial localities. The hematozoon was present in strange forms, and in short, it was completely outside the circle of the known pathogenic microbes. In 1880, the technique of examination of the blood was unfortunately very imperfect, which contributed to the prolongation of the discussion relative to the new hematozoon. At the same time, the endoglobular parasites were discovered in different animals. In 1889, my hematozoon had been found in the majority of malarial regions. Not possible to doubt any more either its existence or its special role, especially the cerebral concentrus, and study its formation in the blood of persons affected by malarial fever. I found leukocytes more or less loaded with pigment, but pigmented spheres bodies of variable size possessing a mirrored movement, free adherent to the red cells, not pigmented corpuscles forming clear spots in the red cells. Finally, pigmented elements, concentric in shape, attracted my attention, and from then on I suppose they were parasites. The malarial hematozoon is a very small protozoon, since it lives and develops in the red blood cells, which in man have a diameter of only microns. That's the ounce of meter. One can summarize the principal ways in which the parasite appears in human blood as follows. 1. Small, rounded, non-pigmented elements. 1 to 2 microns in diameter, forming clear spots in the red cells. Multiplication is by halving or by multiplication. 2. Amoeboid elements inside the red cells or adherent to its surface of variable forms and dimensions containing blackish pigment. Multiplication is by segmentation, and a 3. Crescentic shaped body. 4. Flagella. Siemens, Schodenin, and Siglecki showed that analogous elements are found in coccidia, and they are male elements destined to fertilize the female elements. It has now been definitely proved that the hematozoon of malaria has, like coccidia, two forms of reproduction: asexual, represented by the segmented bodies, and sexual, the flagella being the male elements. I have always defended the unity of malaria and its hematozoon and postulated species described under the names of parasites of tropical malaria, aestophil, atenomial fever, Tachyon, and cardinal fever appear to me to constitute simple varieties of the same hematozoon. In what state does the hematozoon exist outside the body, and how does infection occur? I became convinced that the microbe was already present inside the human body in a parasitic state, and probably as a parasite of mosquitoes. This opinion on the role of mosquitoes considered by the most observers at this time as not very likely. King America has suggested in 1883 that mosquitoes played a part in the etiology of malaria, but he did not know of my work on the hematozoon of malaria and could not specify what part the mosquito played. I indicated clearly the route that was necessary to seek out what became of the parasite in the body of mosquitoes which had sucked malarial blood. Having left the malarial countries, it was not possible for me to verify the hypothesis I had put forward on the role of mosquitoes, and it was to Dr. Ronald Ross, who also received a Nobel Prize on this I think, in 1902, that we owe the demonstration that the malarial hematozoon and the closely related malaria of birds completed several phases of evolution in the Culicae and propagated by these insects. Ross acknowledged in many of his papers that he has been usually guided by my deductions and by those of P. Manson. Today, the transformations which the malarial parasite undergoes in mosquitoes of the genus Anopheles are well known, and no further doubt is possible on the role these insects play in the propagation of malaria. From 1899, my principal work on malaria has had the object of the Culicae in their relations to malarial endemicity and of the relational prophylaxis of this dreadful malady. I have been able to show that Anopheles can be found in all the malarial localities. Dr. Battesti and I have demonstrated that Anopheles can be found in all the malarial localities of Corsica, which have been considered free of these Culicidae. I have been able to write malaria, the history of which not long ago presented such obscurity, is today one of the best known diseases. The study of the clinical forms and the pathological changes has been completed and defined. The hematozoon, which I described in 1880, has been recognized by all writers as the cause of malaria. Its propagation by mosquitoes has been summated, and we now are able to fight the malaria by specific drug treatments and rational prophylaxis. I come now to the examination of my other work. Then this is my description. Gives a long script here for except for the work of the trypanosis. Investigations on the structure of trypanosis and that of the Flagellidea, in general, the agglutination of trypanosis and the conditions which produce it, the differentiations of the trypanosomiasis, the trypanosis of rats, of horses in the Gambia, of sleeping sickness, one affecting bovines, one for new specific phonetic of equinoxes and bovines, of birds, cyclonians, bacterians, or fishes. Trypanoplasma have been found in the rat, carp, and minnow. Their structures differ markedly from that of the trypanosomes as I have ascertained from well-designed specimens. Finally, I should mention a series of papers on prophylaxis and treatment of diseases due to trypanosomes. If doctors for you, I undertook new experiments aimed at investigation whether the combinations of two different arsenic preparations would not give better results than the use of arsenic acid alone or toxil alone. The results we obtained by combining autoxyl with arsenic trisulfide or arsenic iodide have been very successful, and we hope that this new drug treatment may be used in human trypanosomiasis or sleeping sickness, which is raging at the present time along the natives of equatorial Africa, on which there are more and more cases of Europeans. To summarize, for 27 years I have not ceased to busy myself with the study of parasitic protozoa of man and animals, and I can say I believe without exaggeration that I have undertaken an important part in the process which has been made in this field. Before the discovery of malarial hematozoon, no pathogenic endoglobular hematozoon was known. Today, the hemocytozoa constitute family important for the numbers of genera and species, and also for the role of these protozoa playing in human veterinary pathology. By directing the attention of doctors and veterinary surgeons to the examinations of blood, study of the endoglobular hematozoa prepared the way for the discovery of the disease due to trypanosomes, which themselves also constitute a new and very important chapter in pathology. The knowledge of these new pathogenic agents has thrown a strong light on a large number of formerly obscure questions. Progress obtained shows once more how just is the celebrated axiom formulated by Bacon, Bene essere per causis scriere. If you want to know that is. Yeah. Okay. Here we see the mosquito in question, which is linked to malaria. And here we see the plasmodium linked to it. And here you see some of the beautiful pictures, beautiful drawings made by Alphonse Laveran of the parasite in question. Well, thank you for watching. Of course, as always, this is a summary. If you want to read the complete, which is quite a bit longer than the summary, of course, go to the official site. Thank you for watching and see you hopefully next week.